Today what we're going to be working on is a gram staining procedure for bacteria. So the first thing that I want to bring to your attention is that I am already using a pre-heat fixed smear slide. So I have my target circle, my initials, the date, what I'm working with, and what the procedure is on my slide. Depending on what class you're in, you may have different labeling systems, so please be aware of that. So the first thing that you do whenever you are working with a gram stain is that you will take crystal violet and you will add a few drops to the target circle. You're going to wait approximately 20 to 30 seconds, and I'm going to use my watch. You can also use a stopwatch, and let that set on the slide. Your next step after that is to take just regular tap water, and what I like to do is hold my slide at a 45-degree angle and take it above the stain and gently run it back and forth to get the stain off of the slide. Then just tap it off a little bit. Your next step is to take grams iodine and place that on your target circle. So the grams iodine will sit for approximately 1 minute. Again, I am going to use my watch, but you can also use a stopwatch. So if you are using the Benson's lab manual, the next step in their procedure says to directly decolorize. However, I am going to be washing off my slide again with water just to give it a little extra help. So this next step, you do not apply decolorizer directly to the slide. Instead, we're going to do a similar technique with our water bottle, lift the slide at a 45-degree angle, and gently decolorize. So you can see that once I had the decolorizer, more of the crystal violet started to wear off. And also, you might have realized that my target slide has disappeared. That's okay, because the last step is to add safranin for a minute. And it should just dissipate onto the slide. So while that is sitting for a minute, I would recommend getting out bibulous paper so that once we take our final wash, we can blot it dry. So once I've reached a minute on the safranin on the slide, I'm going to take it again, 45-degree angle, and gently wash off with water. You can tap off any excess color, water into the drain. grab bibulous paper, find an open page, and then I like to place it down and close the book. And I'm just going to lightly press on it. Be careful not to swipe it through. If you swipe it through, all of your bacteria will come off with it, and then you won't be able to see anything in the microscope. So once your gram stain is done, occasionally you will be able to see the reddish colored dyed bacteria. That's typically because there is more bacteria there, but other times you won't be able to see what's on your slide until after you get it under the microscope. So that's how you do a gram stain.